The, thank you so much. Okay, so moving on, as we are talking about the courses, so here are the different courses that are there. So this is also what you have access to. So if you are only registered for the program, you will have access to these links. Not everybody may not have access to the links if you're not registered yet for the program. So these are the courses that you get access to from here. And then we have a progress tab. So what does the progress tab do? It shows all of the different progress you have made across all the sites. So this is the education site where you have the courses, the forum, and your own profile. So right now, as we are all starting out today, we are all going to be starting with 0. And as we move on with this program, we are going to see there are going to be different numbers. So once you complete any of these courses, you get 500 points. Similarly, if you run a pipeline, you get 50 points. So all of those points would be added to your progress bar here. And we can see both individual progresses as well as the progress for the entire group or the entire class right here. The second platform that every registered participant has access to is the Server T-Bioinfo platform. So if you have already registered for the program, you may have received an email from marketing@pine.bio with the individual access to this platform. You cannot change the email ID to the Server T-Bioinfo. You will have to use the login ID that we have provided to you. So please make sure that you're able to log in with the ID that is provided. And in case you have any problems, please let us know so that we can make sure you have all the access details before the next session. And the third platform that we have, that we will be using during this program is the Codeomics Logic platform. So the coding platform has courses in R and Python, and we will be helping you learn about applying R and Python to biological data. So these are all the resources that we will be utilizing during this 1-month program. And if you have any questions regarding this, you can either reach out to africa@pine.bio or to marketing@pine.bio anytime. Okay, so if you are not able to access Lydia, so please send us an email with a screenshot so that we are able to help it out, and we'll do that beyond this session. And now to start with today's session, I would like to invite Ilya to take over from now on. Ilya, over to you.
Thank you, Deepsa, and welcome everyone to Omics Logic Metagenomics in Nigeria. We're excited to be starting this program today, and we hope that most of you will be able to benefit from the different resources that we provide as we learn about metagenomics. So let me start by introducing what this program will be focused on. We hope that most of you already have some background in molecular biology, and you will be able to apply your interest in the microbiome, which is a growing and very interesting and dynamic field, to learn how to explore microbiome research projects and look at different challenges that are being addressed using a variety of different techniques. This primarily includes metagenomic sequencing, which refers to high throughput generation of short sequencing reads that could either help us provide a taxonomic description of a particular environmental sample or study in-depth analysis of different genes that are being produced by a community of microorganisms. Now, the program is structured in such a way that we will go from learning about these different applications and techniques to developing an independent research project. So we hope that even today, and as you get started with your learning, you will start thinking about what kind of data, what kind of question, what kind of analysis you plan to do. And by the time you complete this program, you will either have a project proposal, which we will explain how you can continue to work on after you complete the basic training and join our research fellowship program, or even complete some of the analysis in as short as 1 to 2 months. So to get started, let's talk about the history of metagenomics and why do we have these novel technologies that help us characterize the microbiome. So just to give you a little bit of history about microbiology, where the study of microorganisms led to the creation of tools and approaches that are still used today and continue to uncover the veil of a world which we do not see, but yet has such a profound effect on everything around us, including our own health. So here you can see Louis Pasteur, that lived in the 19th century. He's a French microbiologist, a chemist, and the pioneer of the germ theory of disease. Back when he was involved in his research work, he was interested in seeing the veil from the microorganisms being uncovered and really understanding what is the impact of these germs or these microbes on everything around us. And so the way that this first happened, it was through the microscope. In fact, the smallest thing humans could see was about as wide as a human hair. And when the microscope was first invented, suddenly we could see a completely new world of living things that surrounds everything that we touch, including our water that we drink, the food that we eat, and even living as communities on our bodies. And so the first observation of a bacterium was made around 1673 by the Dutch microscopist Anton van Leeuwenhoek, thanks to the microscopes that he had invented. And these enlarged from 50 to about 300 times whatever he was observing. As the results improved, more and more representatives of the unseen world have been discovered. And so you can see here an example of today's microscope. They are tremendously powerful that allow humans to see smaller and smaller elements. You can see here, in fact, a number of phages that are attacking E. coli bacteria. So this is the resolution that we are at today. And so as a result, we have a greater ability to see and understand the world of these unseen microorganisms. But microbiology itself ended up being defined not only by the organisms that it studies, but also by the tools that are used to study them. These tools are continuing to evolve and have already evolved to allow us to study and categorize organisms into our understanding of the living world around us. So technology for bacterial culturing, coupled with a technique to stain bacteria, first improved our ability to isolate, classify, and determine whether bacteria is the cause of human disease and even select an appropriate treatment. But today these tools continue to develop, and we can see a greater resolution into this world of unseen microbes. The discovery of culture media allowed for the development of microbiology in the 19th century. Bacterial culture is an artificial medium that allows growth and isolation of bacteria, and the first to have cultured a bacterium in a reproducible way was Louis Pasteur in 1860. Gram staining is a bacteriological lab technique used to differentiate bacterial species into two large groups, gram-positive and gram-negative, based on the physical properties of their cell walls. Gram stains are performed on body fluid or biopsy when infection is suspected. Gram stains yield results which more quickly than culturing and are especially important when infection would make an important difference in the patient's treatment and prognosis. So here we can see an example of what that looks like. You can see how the bacteria is cultured and then stained. And this ability to stain could be used, for example, on cerebrospinal fluid for meningitis and synovial fluid for septic arthritis. So a wide range of different diseases could be characterized and accurately classified using such techniques. The connection between disease and microorganisms has been established back in the 1800s, but these studies provided not only important information, but they also followed a specific technique that only could look at bacteria in isolation. But it became apparent that most microorganisms actually live in complex communities. Many members of this community are impossible to culture in isolation, And they live and work together almost as a single organ, which we will learn about in just a few slides. So that we know today that they form highly regulated, structurally and functionally organized communities that are attached to surfaces as biofilms with interspecies collaborations as well as antagonisms that contribute to their overall ecological stability. Due to the advances in metagenomic sequencing and other omics technologies, enormous databases of data from such communities have led to the emergence of computational metagenomics, which can help study the 99% of uncultivatable organisms in the context of their natural environment. So this is a brief timeline. In the 1600s, we had the first observations of microscopic organisms. By the 1800s, the connection between microorganisms and disease has been established. From the late 1800s until present, culture, staining, and microscopy were developed and used in both research and clinical applications. In the 1980s until the present, we have the emergence of DNA sequencing and the application of DNA sequencing to the study of microbial communities. In 2008, the Human Microbiome Project was launched by the NIH. What kind of an impact this had on the study of microorganisms. And from the 2010 and to present, bioinformatics and especially multiomics integration have really defined the world of metagenomics. The stability of these communities is fascinating, as many of us probably know when trying to battle such common skin conditions as eczema, acne, and dry skin. Forming distinct communities, these microorganisms are interconnected and determined to maintain their community. Bacteria within a biofilm can communicate with each other by producing, detecting, and responding to small diffusible signal molecules in a process called quorum sensing, which confirms benefit for host colonization, biofilm formation, defense against competitors, and adaptation to changes in the environment. Quorum sensing activities in biofilms are also involved in the virulence and pathogenic potential of bacteria and are therefore an important factor in our ability to understand and control bacterial infections, as they enable microorganisms and biofilms to become more tolerant to host defenses and antimicrobial agents. Inside each community, there is a balance of bacteria, fungi, and viruses that control species abundance and activity. So the human microbiota is estimated to be between 10 to the 13th to 10 to the 14th of microbial cells, with around equal proportion of microbial cells to human cells. Previously, it was even thought that that number is much greater, but today these figures have been adjusted since 2016. These numbers are actually derived from the total bacterial cells in the human colon, which is approximately 3.8 to the 10 to the 13th of bacteria. The organ, the gut, harbors the greatest number of microbes. However, each one of these microorganisms carries important functional properties in genes that contain genomic information about different functions that our body cannot do on its own. These properties and the genomic information about microorganisms have been of a growing interest to the scientific community, resulting in well over 80,000 publications in the course of the past few years. The majority of the studies have focused on the intestinal microbiome. So here you can see how the body is dominated by specific types of microorganisms depending on the organ. And what we are discovering today is that these microbes actually communicate with each other and form almost a single organ that functions in a variety of ways, supporting our immune system, digestive system, and controlling the progression of disease. In fact, not so recently, in February 2020, a new review article came out on the gut-skin axis, which summarizes the current knowledge of the interrelationship between microbial dysbiosis overall, including the gut microbiome, and skin conditions. And it describes how inflammatory and microbial influences between the gut and skin for a healthy state and a dysbiotic state are a result of a variety of microbial changes. So this dysbiotic state is characterized by an impaired gut barrier, imbalance in the gut microbiome, reduced mucus layer, reduced secretion of different antibodies, barrier disruption, intestinal permeation into the bloodstream, and gut inflammation, and an impaired skin barrier, imbalance in skin microbiome, reduced human and microbial antimicrobial peptides production, skin rashes, thickening lesions, and skin inflammation. And we can see how gut and skin dysbiosis are connected through an immune imbalance. So in this study, you can clearly see not only the role of the skin microbiome, but the relationship between the skin and the gut dysbiosis in their relationship with the host. So it's really a very close and interconnected organ. Now, to study this, there have been a number of microbiome studies that have significantly advanced through major initiatives such as the Metagenomics of the Human Intestinal Tract and the subsequent Human Microbiome Project. These are huge studies that have been made possible through the advances in metagenomic sequencing, which offers a cheap alternative to study microorganisms in their community without the need of culturing and with rapid turnover. Each new project here represents vast opportunities and challenges to determine which microorganisms play key roles in the development of certain environmental conditions needed for healthy function of human organs and which can be harmful to it. Specifically, we now know of the gut, skin, lung, and even brain microbiota that have been characterized, and new associations have been found from mild conditions, such as sensitivity to different types of foods, to very acute conditions, such as the development of Alzheimer's disease, or even susceptibility to a variety of infections. Over the past decade, more than 1.7 billion US have been spent on human microbiome research. Major projects are underway in the United States, the European Union, China, Canada, Ireland, South Korea, and Japan. This investment has confirmed the significance of the microbiome to human health and development. Some 20% of US investment in such research has gone to two phases of the Human Microbiome Project, which is creating the research resources needed to study the human microbiome. One of the main goals of the HMP, launched in 2007, was to create not only a data set, but also a toolbox of computational techniques, analytical methods, and clinical protocols so that others can leverage this to understand the role of the microbiome. So what is known so far about the microbiome function is that it plays a significant role in the differentiation of the human immune system. Oh, sorry. Let me start that over. What is known today about the microbiome is that it plays a significant role in the differentiation of the immune system, food digestion and nutrition, energy generation and conservation, metabolic regulation of fat storage, processing of environmental chemicals, maintenance of skin and mucosal barrier function, as well as prevention of invasion by pathogens. So what we will do in this program is we will try to identify how such microbiota could be studied using next generation sequencing. So the essential steps here are to understand when we have a community that can be comprised of millions of individual microorganisms, how can we use an effective approach to sequencing the genomic information about these communities to characterize the composition, dominance or species richness, and the interaction between the microbiome and their environment, forming specific conditions that have a major contribution to our human health as well as the environment. So what is the study of metagenomics? Unlike other omics, metagenomics does not study a single organism, but the whole community at once. So it provides us with a true snapshot of a collection of organisms that are specifically in the process of interacting with their natural environment. Since the genomes are so diverse, This is typically accomplished by sequencing a specific marker portion of the genome instead of all of the genomic material present in a given sample. So specifically, this is referred to 16S metagenomic sequencing, where the amplification of the ribosomal RNA from the smaller subunit of the ribosome that is specific to bacteria can be amplified using PCR. The 16S gene sequence contains hypervariable regions, also known as HVRs, that provide species-specific signature sequences that are useful for bacterial classification. The benefit of amplification is tied to the nature of sequencing read length, which can maximize effective output from very affordable sequencing technologies. So the way that this data is annotated and analyzed is through computational processing, which are called metagenomic processing pipelines. So those are usually utilizing something that's called operational taxonomic units, or OTUs, which can be used to classify groups of closely related individual reads. These OTUs are formed into groups of closely related genomic sequences and represent essentially a cluster that could be assigned to a phylogenetic clade on the taxonomy of the full bacteria that is present in a sample. The way this is done is by annotation using different types of databases. These databases include such repositories as the SILVA, Green Genes, NCBI, and RDP. And so in the context of this program, we will learn how to utilize these reference databases to annotate and understand what kind of composition are we looking at in a given microbial sample. So ultimately, based on that mapping of specific regions amplified from the 16S ribosomal RNA gene in the small subunit of the bacterial ribosome, OTUs can be assigned to belong to a taxonomic class of bacteria, yielding important information about the proportionate view of the community. Each community can then be studied by comparing with others or between similar communities in different conditions or individuals. Such analysis includes an understanding of how the different types of sequencing techniques and analysis of sequencing metadata and data could be combined together to really understand the microbiome. Now, sequencing is not the only technique that is used to study the microbiome. Another technique is called mass spectrometry. Sequencing allows us to either understand composition or study specific isolated genome. And mass spectrometry can tell us what types of proteins and metabolites are being produced to understand what functional contribution each species might have, or what types of products a community produces and how it is metabolized through this communal analysis. So many questions about the role of microbiome members and communities are being answered using these technologies. And we think that the next phase of microbiome research lies in the ability to integrate multi-omics datasets that combine next-generation sequencing, mass spectrometry, structural data, as well as phenotypic information about overall changes in the community and their environment. So many questions are going to be answered through such integration, as you can see represented here, through both annotation of functional pathways and function, as well as unsupervised integration between different types of microbial taxonomy, different products that they produce, and the interactions between them that could be studied using sequencing. So in this program, the Omics Logic Metagenomics program, we will go through different examples with associated training and practice, hands-on practice on how to use such techniques to be able to analyze data and interpret the results of the analysis in a meaningful way. So to get started with this process, I want to show you several courses that we have developed for your asynchronous use. Now, as a part of this program, this will be supplemented by hands-on activities that we will do in group sessions For those of you joining that have not signed up for the program, you can all try out this Introduction to Metagenomics course to really understand how this works. So let me take you to the platform and show you what is inside these courses. So this is the link to our organizational page. Again, if you're already a part of the organization, you could be just going to this organizational page, and I will show you where to find the associated coursework. So for those of you that are already a part of the program, you would go right here to the Courses tab. And once you're in the Courses tab, you will see the 3 courses that we're going to be covering in this program, which includes an Introduction to Metagenomics, Metagenomics 1, and Metagenomics 2. So these courses have both a theoretical review of practical assignments and some data sets that you can go through to understand how the microbiome functions. So here, if you go to the course tab, you will see Introduction to Metagenomics, Metagenomics 1. So you can start working on those courses. For those of you that are not a part of the program, you can just go right here to courses, and you will find these courses outside of this group. So you want to look for Introduction to Metagenomics, which is a free course that will be available to all of you. Okay, so Introduction to Metagenomics right here. Let me just paste the link here. Okay, thank you, Beepsa. You already pasted Introduction to Metagenomics. So what is Introduction to Metagenomics? If you go to the Curriculum tab on the Introduction to Metagenomics, you will see here that we describe types of metagenomic sequencing, analysis of 16S amplicon metagenomics data, next generation sequencing and analysis of ribosomal RNA gene sequences, taxonomic classification, as well as the impact of microbes on the human health, the gut microbiota, the brain, and others. So this provides a very thorough overview of a lot of the things that we covered in today's session. After completing this course, you will receive a certificate of completion that you will be able to leverage in building your resume and your capabilities as a bioinformatician. So in other courses that we will go through in this program, you will find additional hands-on applications and important examples that we will go through. So the way these courses are organized is that you will see the expectations for each one of the activities, mapping of conceptual topics, because it can get quite confusing with a lot of terminology and a lot of very specific details that you have to know to sets are being analyzed, for which reason and how. It will introduce you to a variety of data sets or projects that you will be able to work through and replicate analysis that was published and described in high impact research articles and journals. And then those analysis pipelines will become an area for you to practice so that you can understand the analysis, not only by reading about it, but actually doing it yourself. As a result, you will accumulate a number of points that will result in levels and badges that you will get for completing certain activities, certification through our online asynchronous point system, as well as in the context of this group or of this program, this training program, you can also get a certificate of completion or a certificate of excellence, which will go to those that actually apply these methods to their independent project. Now, in these online modules, you will see how this key terminology from a bioinformatics perspective is linked to examples of projects where you can analyze different types of conditions that both represent a clinical condition and some other examples that are not related to human health. So what are some of these tools that we will cover? In the Metagenomics program, we will rely on the tBioInfo Big Data Analysis Platform that contains specific analysis pipelines or processing tools to manage and analyze data from next generation sequencing. So here you can see an example of a popular pipeline called KIME2, as well as the data to pipeline. So these pipelines could be used to both process the data, eliminate the necessary junk or the noise from the data, as well as do downstream analysis and annotation. So to give you an idea of how this works, let's go to server.tbioinfo. So Bipsa, thank you for posting that link in the chat. Please let me know once you are on this platform so that we could take a look at some of these example pipelines. And for those of you that do not have an account yet with us, you have not received the email, we also have a demo account that you can use that works perfectly fine. It just does not allow you to upload your own data. So Bipsa, if you could paste the details. And for all the participants, please do let me know once you're on the platform and you're logged in. All right, thank you, Nita. Anyone else? You need to go to server.t-bio.info and either use account details that you received by email or use the demo account with the username and password that Bipsa posted in the chat box. Okay, so as you're logging in, I'm just going to give you a little overview of what you are seeing here on the screen for areas of analysis. So first of all, you will see that there are multiple sections for sequence alignment, or sorry, for sequence analysis listed out for each individual section. So each section represents a collection of methods required to process or analyze data. Under some of them, you will see this drop-down arrow that represent the demo pipelines that you can run. Now, these could be run both with your account as a part of the program or a free demo account that you might have. So we will take a look in just a few minutes on how to do that. Then you see right here your previous pipelines. So what are the pipelines that you tried running or completed in the past? You will see right here your profile and data management so that you can understand what is your account and how you can upgrade. And you also will see here some data mining, utility scripts, and machine learning sections. So we will cover today a number of examples just to understand how they work and what are the inputs and the outputs. And this will include whole genome analysis, where we can map whole genomes onto a database of known bacterial genomes. KIND2 is an example that I will show you as one of the most probably known pipelines for analysis of 16S ribosomal RNA sequencing, and the data to amplicon pipeline, and what are some of the ways to annotate and interpret this data using a popular phyloseq package in R. Okay, so let's go to the platform. And the first section that we will go through is this section for Metagenomics Microbiome. So if you click on this arrow, you see here Demo Metagenomics Regular. Okay, so we can go in there. And the first thing that you will see is this pop-up that explains what the pipeline can look like and what does it do and what is the meaning behind each one of the buttons and how to really use them. Okay, so you can close this for now. And now we will take a look at one example pipeline that simply takes in some data from the American Gut Project, which is the largest crowdsourced citizen science project to date, because it involves many people from a variety of backgrounds that have contributed their samples of saliva, fecal samples, and others to understand microbial composition between a wide range of people. After you've selected this, you click on OK. The second thing that we will do is we will map onto a reference database using bowtie to align. So clicking here, you will see that we are mapping on a database called Silva. And this is a database of 16S sequences. So here you can click on OK. And then we will create an abundance table using this annotation method called pause count, which looks at the count of reads that map onto a particular entry into the database. So here you click on OK. And then we want to annotate the positions in the 16S ribosomal RNA database to understand abundance of particular regions and how we can see those in our output files. So here you can see how we create a 3-step pipeline. And then we click on end, and we will see what are some of the outputs. Okay, Nii is saying that the website is not responding. Could you clarify what is not working for you? So please put it in the chat if you have some issues with building this pipeline, and we will try to see what the issue might be. So after you've done this pipeline, you will notice on top, download pipeline output files. So you can click on this and see some of the results. You will see mapping statistics, which is a process of annotating how many different reads mapped onto the reference. You will also see the result of all of our bow tie mapping. And then you will see a table after annotation, which is what we want to look at. So let's click on table after annotation and see the file that we downloaded. So I'm just going to open this in Excel and show you what's inside. Okay, how many were able to generate this file? How many people were able to generate the result file? Okay, so if you have, what you will see is that after we have the ID, okay, so we have here the ID of this particular 16S ribosomal RNA entry, and then how it is organized by taxonomic classification. So here you can see K, P, C, O, F, G, S, right? So these are taxonomic levels. So based on these taxonomic levels, we can now organize, okay? So here you can see the taxonomic levels. How we took 16S ribosomal RNA sequences, and we can classify them by species, genus, family, order, class, phylum, or kingdom. And now that we, okay, so Kehinde, just to kind of remind you what we did on the platform, I'm just going to show one more time. You go to areas of analysis. Okay, again, if you're not able to log in, please use the demo password and email account that Beeps provided. But here you will find, once you're in, a drop-down right here next to Metagenomics Microbiome. You can also hover over this little eye to really see what each section does. So under Metagenomics Microbiome, we want to take this first demo, Metagenomics Regular. Once you're in here, you will see a pop-up which you can close. And in the future, if you don't want the pop-up to come up, you can click on this dashboard. Then you click on Start. You read about the input data that we're using, which is from the American gut sample. bow tie to align, where we describe the database that's used for mapping, pause count, which creates an abundance table and calculates how many reads are present for a given entry into the database, and then annotation, so that we can annotate based on taxonomic classification. So after that's done, we click on End, and then you should see download pipeline output files, and you can open this folder like that. And now you see a number of different elements coming out in this output file folder. So in this folder, we managed to download a file that looks like this. In this file, you will see that the result, this is just one sample. So this sample has a variety of different positions, and we can sort by this column to find the biggest, the most abundant entries into the database. And so in this way, we can start thinking about what kinds of bacteria are likely to be present in the sample, and how do we understand the impact of such bacteria on the phenotype, which is described by metadata. Now, in this case, this is a manual pipeline where we take the downloads as a table, and then we can visualize and annotate this using a variety of different annotation techniques. But let me now show you a different example where this process is automated and is specifically designed for sequencing of hypervariable regions. So to do that, I will go into this last section called Amplicon 16S, 18S. And I will use this example project for Metagenomics Amplicon gut-brain axis. Now it's called Amplicon because we're amplifying hypervariable regions and then annotating those hypervariable regions again on the same database, but only looking at the regions that are most representative of this taxonomic classification. So here you can see the process of amplicon sequencing with PCR amplification of variable regions. Then you can see what the data to pipeline consists of, which is primarily the workflow to reduce noise in our input data. So the reads are denoised, merged by pairs and sequencing, and then organized into clusters of operational taxonomic units. Again, those are reduced from chimers or chimeras, assigned taxon by looking at the database, and then we can build a phylogenetic tree that can actually help classify differences between microbiota. So here we use an R package in Phyloseq that is going to be used for visual outputs of this analysis. So once I get to download pipeline output files, I will see a number of different outputs, including some plots, which are given in a PDF format. So for example, here we see air plots, quality profile plots, as well as Phyloseq plots. So we can download the Phyloseq plots to see what is the composition between different samples. So here in Phyloseq, we see a family level taxonomy that is distributed between a number of samples, 6 samples, BKF01, 02, 03, and then 13, 14, and 15. And here we can see the composition or the species richness based on this family taxonomic organization. So then we have to look at how not only are they individually represented, but also how are they interrelated. A species that is more divergent, farther away from another species, is more interesting than a species maybe that is closer together, because they will have a wider range of functionality. And so we build this tree of operational taxonomic units, and we can actually study the different types of diversity using additional methods like Chow and Shannon to understand how species diversity, as well as species richness, can play a role in a particular phenotype. Okay, so now we have a question. When opening TXT file data in Excel, is it best to save it as XLSB format or later retrieval? Okay, seems like a question that's not directly related to what we're talking about, but yes, overall, you can probably save it as an Excel file. That way you'll be able to use it later again, easily opening it up in Excel, and it will include saving any kind of figures that you have in your data that will be saved as well. Okay, so in this example, we're using a particular data set. So to give you an idea of some of these data sets, I want to introduce a few different applications of the microbiome that we will use in this program. But before I do that, I also want to mention that we will use some of the methods that we've seen here, including phyloseq and the different methods that are packaged within this package, as a way to learn a little bit more about coding. So now to see an example of how we will leverage these examples in metagenomics to understand coding and popular packages in R and Python, let me take you to another resource that you will gain access to in the context of this program called codeomicslogic.com. Okay, so if you could please put that on the chat. Thank you, Aditya. So please go with me to codeomicslogic.com. And here you can register for free. So this is a free account that you can generate. And I will explain in a couple of minutes how this works and how we will use this for generating figures that you see here on the screen. Okay, and for Nita's question, we will learn about the Simpson index and the Shannon index. They have a different ways of calculating differences between the sample composition, but those specific differences could be interpreted in different ways depending on the type of input data we have and depending on the question that we have. Okay, so let me know once you are logged in or you have created an account on code.omicslogic.com. Okay, if we could again post that link in the chat with HTTPS, it will be a link. Thank you, Bipsa. Okay, so let me know once you're on your account on codeomicslogic.com. All right, I see some people started logging in. All right, great. Thank you, Nita. All right, thank you, Jeremy and Lydia. All right, great. So once you're in the account, you will find a course called Metagenomics. So in this course, you will see some of the examples of what we will be using to amplify our understanding of the microbiome composition and species richness, but also learning about some very basic functions in R. So in this course, you will find how to generate an understanding of what types of bacteria are present in a sample by proportion or by abundance, and what are some of the typical considerations around leveraging the different outputs from the pipelines that we already tried, so that your understanding of the composition is also considering the abundance of input data that you have and the sequencing depth that you are using in maybe a different set of samples that you're using overall. So in this course, you will find different practical tutorials. And let me just show you how those work. Okay, so once you're here, you can see here a variety of other courses that you obviously will get access to as well. But here, let's just take a look at this example. And we'll go right here to number 4. And so in this course, you can see that we start with an introduction of a data set. This data set includes a variety of different conditions. And as an example, we can use those that exercise rarely and those that exercise regularly. Okay, you will find in the source data set, which is linked right here, a variety of different metadata that also include how often people eat, how often do they drink, what is other kind of lifestyle conditions that we could link with microbiome composition. And so once you read about this, let's go to this practice yourself. You cannot open this unless you actually complete the previous steps. But here you'll see an example of how does this actually work? Okay, so just by simply clicking on this run button, you will be able to try out the code that is here. And so obviously you will be able to modify the code as you try to arrange this data in such a way that you can prepare it for a publication or present it to your peers comparing composition by different taxonomic classes between different conditions. Okay, so here you will find both an explanation of the challenge, the code basics, as well as a practice playground where you can try to modify this code and produce the types of results that you would like to use in your publications. Okay, so what is going to be included? What we'll do is we will look at a variety of methods as well as an understanding of the typical considerations behind the statistical analysis as well as the machine learning methods that are typically used in analysis of omics data, as well as specifically for metagenomic analysis. And we will go through very basic things to learn how the code works, as well as very specific applications of the most popular packages that are used in metagenomic analysis. So let's talk about several examples that we will use as data sets to practice. So applications of metagenomics include analysis of skin conditions, food sensitivity of the gut, autoimmune disorders, cancer, antibiotic resistance, drug resistance, and even ecological and agrobiological studies. So we'll take examples from skin and acne, sensitivity of the gut microbiome, and these different conditions to play around with the different methods of analysis and understand that in the context of different types of questions and data sets, we should use appropriate methods to process and annotate as well as visually present our data. As I mentioned, one of the first projects that we'll try is from the American Gut Project. So the American Gut Project is a citizen science project that has a lot to offer for those interested in basic and microbial composition of different types of samples from healthy and mildly sick individuals. Here, we will learn how to create a report similar to what you see right here for an individual that submits their data, and also how to group people by different types of composition of their microbiome to study a condition. However, in this example, it is very difficult to disassociate the impact of microbiome from other factors that are not related, such as behavior, comorbidities, and other unreported data that we have associated with each sample. So to study the effect of microbiome changes in a unique way, we can turn to a project that we will use that tries to characterize the relationship between the gut and the brain axis by studying microbiome and behavioral anxiety. So here we will see how changes in the gut microbiota in mice through antibiotic treatment followed by a gavage of donor stool from other mice can help us determine if significant behavioral changes are induced and anxiety levels are associated specifically with microbiome that comes as a result of high-fat diet or a regular diet for mice. Such studies are really important for our basic understanding of the role of the microbiome, as well as in the translation of different types of probiotic, prebiotic, and transplantation strategies for treatment. But we will also look how microbiome, or the kind of microbial community that we're studying, can have a variety of other types of effects. And for example, we will take use cases from tuberculosis, Ebola, and even rhizobacteria, which is a bacteria that grows on the roots of plants, and how to study such data. Importantly, we will see that the microbiome is now considered to be a major player in many other areas of biomedical research. For example, screening for diagnostic signatures in precision medicine, and even association with treatment efficacy. Or the therapeutic considerations for microbiome transplantation to study such conditions as Alzheimer's disease. So here you can see an article where China has already approved Alzheimer's treatment that targets the microbiome through transplantation. And so importantly, we will not only look at processing individual types of data, but we will look at how bioinformatics plays a major role in our ability to organize, analyze, process, and find associations between clinical and omics data, or phenotypic and omics data, to find important translational insights about diagnostics, therapeutics, and even agrobiology. Because today we know that the microbiome in agriculture is considered to be an important component of production of different crops, resistance and resilience of crops on harsher conditions that are caused by the climate change, as well as the ideas about how to develop novel interventions and provide better security for food around the world. So here we can also see how the applications of microbiome play an important role in what's going on right now during the pandemic. Many studies have turned to microbiome's role in susceptibility and immune response to COVID-19. And this is an important part because the microbiome is one of the easiest things to sample in the human body. And so this provides us with a unique insight into a variety of fields that have a direct impact on our lives. So in the context of this program, we invite you to master metagenomics data analysis. So after you complete the program, you should be able to find, analyze, and interpret metagenomic data on your own. And you will be able to gain experience by doing this in the context of our interactive sessions. And hopefully, if you have the time to present your project proposal, to also do this to our community. So now I want to open this up for any questions that you might have. I do want to mention that Introduction to Metagenomics and some of the coursework on the codeomicslogic.com coding playground are accessible for free for those of you that have not yet registered. For those of you that have registered, you have access to a lot of additional premium resources that will be provided in the context of this program. And for those of you that have registered, I want to recommend that you already start building up your profile from starting to complete with your full name to adding a photo or an avatar, and then starting to accumulate points so that you can get certification for the different resources that you completed and also gain recognition for the work that you do. Again, 2 types of awards will be at the end of this program, a certificate of completion or a certificate of excellence. And so as you think of your questions, I will just briefly explain what these 2 mean. The certificate of completion will be awarded to anyone who has over 3,000 points, which equates to about 4 different courses, and obviously some practice with the pipelines that are required as a part of the course. Certificate of excellence is awarded to anyone who has over 10,000 points. So 10,000 points is an easy goal for someone that is going for an independent project. So if you are willing to not only learn about metagenomics, but also find the data set and try to analyze this data set and understand what this data set says about microbial composition, you are likely to find yourself easily achieving this 10,000 point goal because your practice with the coding and with the different types of pipelines, those will likely to end up quickly accumulating into 10,000 points. Any questions? So if you have any questions, you can put them in the chat box. Is there anything else that someone wanted to add, Adetayo or Bipsa? Okay, how much time is required per week? We will have 2 sessions per week. Each session is going to last between 60 and 90 minutes. These sessions are recorded, and you can use the recording to review the specific things that we have covered, or also to practice on some of the practical pipelines. For those of you that are interested in joining this and following this in a synchronous way, you can actually take a look at how you can schedule this on a flexible basis. We have all of the information, and I will just briefly show the information here. This information is explained on this page. Okay, so if you want to learn about the details, you go to the top menu right here, and you can go to Omics Logic Metagenomics. Okay, so in this program, I will just take a look at a couple of events to just clarify the schedule. Now, is it required to know Python or R? It's not required because we have these tutorials that you can follow and learn from the very, very basics. But unlike most programs out there that focus a lot on becoming proficient specifically at coding, we prioritize the logic of analysis. So you know how to critically think about your data and use the appropriate tool to be able to answer the question at hand, specifically with the type of data that you are dealing with. So you're more likely to quickly get to the analysis result rather than necessarily going through all of the training to be able to apply this to any type of data. And this is for people that do have some background in life sciences. So if you do already have an understanding of the microbiome, an understanding of cell and molecular biology, you understand what to look for and how does this tie in with the overall concept of metagenomics, this is going to be a huge benefit to you. So again, here you can see the paper, organization page, and you can see the upcoming sessions. So I believe this time is in Western Africa time for Nigeria,
yes,
because
yes.
yeah.
Yeah. So just to address a few questions here, Ilya. For Kehinde to go first, yes, the video of this session would both be posted in the forum as well as this was a free session, the first session. So this will also be available on the Pine Biotech YouTube channel as well for you to review again. If you are already registered for the program, you will be able to find the video posted in the forum, like the topics which I had shown you earlier. It would be posted there, and I'm sharing the YouTube link right now in the chat. So this is a YouTube live session. So you will also be able to rewatch the session again via that link. And there is another question which I would like to address regarding the access to server tbioinfo. So please make sure whenever you're logging into server tbioinfo that there is no space in between your username or password, before or after the username or password, as it is case sensitive. So in case there is a blank space, it will treat your password as incorrect. So if you have already received your details, or even if you're utilizing the demo details, just make sure that you're not putting in space so that you avoid the login in mistake. Yeah, so Nee, if you decide to take up a project and analyze data sets, necessarily the time period of this program is 1 month, but we do understand that projects take a longer period of time. So what you can do is that you can also go ahead and join our research fellowship program, which is primarily dedicated to helping you develop a research project of your own under the guidance of a mentor. So we will talk more about that in the upcoming sessions. So what we would want to do in this 1 month program is to help you understand, get the training, understand the logic, as Ilya has been explaining to you. And then once you're familiar, at least come up with a research proposal, like figure out what your project would be. So we will have a step-by-step process that we will take you through, starting with the training with basic courses, getting familiar with the different pipelines, learning to utilize R and Python for some of the analysis and visualization, and then thinking of these steps in terms of a project that you would want to explore. So if it is not possible for you to do it in 1 month, because that is quite a short period of time for doing your own project, but first getting acquainted with this system and then working on a project, which we see that a lot of our students and participants have been able to do it under 3 months as well. So I will be sharing some examples as well here in the chat with all of you. These are from participants as well as research fellows who are using the account. So the free login is only available for a limited period of time, and you cannot analyze any kind of data sets with the free login. You can only run the demo pipelines. Any pipeline which is marked with a demo, you can only utilize that. For participants, you have educational access, which means that you have 5 terabytes of space for running the pipelines, and you will have access during the entire length of the program, and you will be able to analyze data sets from public domain repositories like NCBI, GEO, and other such public domain repositories. If you are looking forward to work with your own data, we do also offer a research license that you would have to take up the licensing for separately. So for demo accounts, you cannot use or upload any files. There is no option to upload files. For educational accounts, you can use, again, public repositories. And for research accounts, you can utilize a platform full. So these are the different licensing options which are available. Are there any other questions which we can answer? And for any questions, you can either reach out to the African team at africa@pine.bio, or you can also reach out to me at marketing@pine.bio, and I will be able to help you out with these questions. And also there's a cost to join the program. So the total cost to join the program is 150 USD. And we had like a 33% discount for everybody else who had been joining till now, which made the cost only 100 USD. So if you are interested, please reach out to us via email, or you can also put in your emails in the chat box and we will get back to you. So thank you again for joining today's session.
Let me just add to that, we do have a scholarship program designed for those that are eager to learn and apply what they learn to a research program, a project, sorry. So again, this is a scholarship opportunity. It is not for everyone. It is only for those that are interested to develop an independent research project and work on that project until completion. You can get up to 50% off. In special cases, we can consider even bigger discounts, but it is specifically designed for those that are already in some kind of an academic institution would like to utilize these tools for a research project. So if Bipsa, you could post the link to the request of a scholarship, thank you. This link will offer you a consideration about whether that program is right for you. There are certain requirements to connect with your faculty and get recommendation letters, etc. But we do encourage you, we do have another week to complete your registration. If this is something that you're interested in, please follow this process, and we will be, our team, either in Africa or our core team here at Pine, will reach out to you with any kind of follow-up questions. Thank you, everyone. It was great to have you with us. Again, this video will be posted on our social media as well as on this program account. And we're going to be looking forward to seeing you again soon.